Today, we will look at the early 20th century science of genetics by exploring the example of human eye color. We will discuss how the pioneering work of Dr. R.C. Punnett and the Hardy-Weinberg law of equilibrium have shaped our modern understanding of this field. Let's begin. Exploring the early 20th century science of genetics and the Hardy-Weinberg law of equilibrium, we find that genetics is the branch of biology dealing with the transmission and variation of inherited characteristics in particular chromosomes and DNA. Evolution is the gradual directional change of these characteristics, leading to more advanced or complex forms. This understanding of the interaction between genetics and evolution is essential to comprehending the workings of both. British scientist William Bateson and R.C. Punnett gave a lecture in London to explain developments in the field of human genetics. Instead of typical examples of Mendelism such as flower or cotyledon color, Punnett delved into more complex topics such as the Hardy-Weinberg law of equilibrium, using the example of human eye color. The development of genetics has seen rapid progress in the early 20th century. Due to the use of Mendelian inheritance, an example of eye color in humans roughly approximates the F2 generation phenotype ratio of 3:1 and the genotype ratio of 1:2:1. Additionally, this example is used to explain the Hardy-Weinberg law of equilibrium, which was proposed by Dr. R.C. Punnett in early 20th century London. The Hardy-Weinberg law is a fundamental principle of population genetics, first formulated by geneticist G. H. Hardy and physician W. Weinberg in 1908. It states that, under certain conditions, the frequency of alleles and genotypes in a population will remain constant over time. Those conditions are that there is no migration, selection, or mutation, and that the population is infinite and randomly mating. The law essentially describes the frequency of different alleles and genotypes in a population over time. Hardy-Weinberg law of equilibrium is a significant element in genetics and population genetics, allowing for the calculation of alleles frequencies in a population. Dr. R.C. Punnett employed this law to illustrate the transmission of human eye color in his address to an audience in London, as it is based on factors such as random mating and the lack of selective pressures. The Hardy-Weinberg law explores the genetic equilibrium of alleles between a population. This law, introduced in 1908, is used to study genetic variation. This slide shows an example of a hereditary human trait, eye color, and how it is passed down through generations. Here, the dominant and recessive alleles for the trait are represented by B and B, respectively. We can see that when two brown-eyed parents are crossed, in the first generation, the ratio of brown-eyed to blue-eyed individuals is 3:1. In the second generation, the ratio is 1:2:1. The Hardy-Weinberg law states that if there is no outside selection pressure, the ratio of alleles in a population remains the same from one generation to the next. This is expressed mathematically as P plus Q equals 1, where P represents the frequency of the dominant allele and Q represents the frequency of the recessive allele. This is known as the equilibrium frequency. When the frequency of either the dominant or recessive allele in the population changes, the equation is no longer in balance and evolution occurs. The three possible genotypes at Hardy-Weinberg equilibrium are P2, 2PQ, and Q2, with P2 being the most common and Q2 the least common. This knowledge can be used to make predictions about the evolution of different populations. The Hardy-Weinberg equation is a mathematical model which assesses the allele frequencies in a population taking into account evolutionary forces. There are 5 of these evolutionary forces that the Hardy-Weinberg equation looks at: migration or gene flow, genetic drift, mutation, recombination, and natural selection. Each of these phenomena impacts the gene distribution in a population, potentially changing the phenotypic distribution and resulting in evolution of a species over time. Genetics plays an important role in the process of evolution. The Hardy-Weinberg formula states that the population of a species will remain constant without evolution. There are 4 distinct processes involved in evolution: mutation, recombination, natural selection, and isolation. Mutations are random changes to genetic material caused by external factors or errors during replication. Recombination occurs when genetic material is rearranged during reproduction. Natural selection is the process through which favorable traits to a species in a given environment are passed down from one generation to the next. Isolation involves the separation of a species into 2 or more distinct populations that no longer reproduce with one another. All these processes work together to drive the evolution of species. Mutation is a driving force in the process of evolution, introducing genetic variability into a population and providing the raw material for change. As the only mechanism that supplies the genetic variation needed for successful evolution, mutation involves sudden, heritable changes in the genetic makeup of an organism that remains stable. Mutations can lead to evolutionary change in 2 ways. Adaptation, in which changes created by mutation offer survival advantages, and adaptability, in which the change provides a broad range of genetic variability to allow for future adaptation. Adaptation is a process by which a population of organisms adjust to their environment in order to better survive and reproduce. In the early 20th century, Dr. R.C. Punnett provided an example of how adaptation occurs when discussing human eye color. He showed that homozygosity, having 2 identical alleles, of a particular genotype within a population can be the best adaptation for that population in that environmental niche. The Hardy-Weinberg law of equilibrium is a result of this tendency for populations to become homozygous in order to adapt more efficiently. The Hardy-Weinberg law of equilibrium has been a cornerstone in evolutionary biology for more than a century, and together with the idea of adaptability is essential to understand how species can survive and thrive in a constantly changing environment. Adaptability is key in a world with fluctuating environmental conditions, as without it, a species can become homozygous and more easily vulnerable to extinction. It is vital to understand how adaptability works to guarantee the ongoing survival of any species. Genetics in the early 20th century provided us with an understanding of how genetic variation is formed. Mutation is the ultimate source of genetic variation, and over a long evolutionary period, has been either selected against or not highly adapted. The Hardy-Weinberg equilibrium law is a powerful tool to further investigate the genetic components of a population, allowing us to gain a better understanding of the genetic variation that exists within a species. In genetics, gene mutation is a key concept. It is a change in the chemical makeup of the gene which occurs naturally. However, in recent years, the rate of mutations has drastically increased due to the presence of mutagenic substances like 5-bromouracil. This substance is very similar to thymine, which is a nucleobase found in DNA. Mutagenic substances have a significant impact on the genetic traits of an organism and opens up new possibilities for research. Recombination is an important mechanism in evolution. It consists of combining the genetic information from both parents to generate novel and exclusive combinations in the progeny that were not present in either parent. The main source of variations in genetics is mutation, which builds a varied collection of genetic alterations. Then, with recombination, we can observe the many gene combinations that can determine the difference between survival and extinction for a population. Charles Darwin first described natural selection in his seminal work On the Origin of Species, published in 1859. Natural selection is the evolutionary process by which beneficial traits are passed down through generations, enabling organisms to better adapt to their environment. This works on a genetic level, as advantageous traits are passed on through parent to child, increasing in prevalence in succeeding generations. Natural selection results from the ability of organisms to adapt to their environment, and that adaptive ability is inherited through succeeding generations. Those organisms with the greatest adaptive capacity are more likely to survive, and their adaptive capability is then passed down through generations. Charles Darwin's contributions to the sciences, particularly genetics, were integral in the early 20th century. He suggested that species are formed over time due to heritable traits and their aptitude to adapt to shifting environmental conditions, proposing the concept of natural selection. His extensive research completely modified our interpretation of how species can survive and thrive. This slide provides an example of natural selection. For instance, iguanas have evolved through natural selection with terrestrial habitats and carnivorous diets. This is just one example of how natural selection has shaped how organisms live and adapt to their environment. Industrial melanism of the 19th century is an example of natural selection where the melanic form, or dark form, was noted to be more viable in laboratory conditions. Environmental changes meant that its frequency increased significantly in the population, causing the genetic constitution of the population to evolve to better fit the new environment. Experiments were conducted to understand the adaptive value of this phenomenon. Adaptive value is a significant concept in genetics, examining the gain an organism possesses from certain genetic features. Put more simply, it is the genetic edge that organisms receive from their hereditary qualities. This is showcased in the case of human eye color, wherein specific genotypes can possess an advantage over others. The Hardy-Weinberg law of equilibrium is likewise an idea in genetics, examining the frequency of endorsed genotypes inside a populace. This law clarifies why populations that display two or more varieties may be thought of as polymorphic or genotypically extraordinary. This suggests that the populace has more alternatives and provides more adaptive value in terms of increased adaptability. To exemplify, we can look at human blood type, with A, B, AB and O varieties all giving an alternate advantage. By grasping adaptive value and polymorphism, we can comprehend how genetics works and its influence on our health and well-being. Isolation is an essential component when discussing evolution, which is the separation of two or more populations to deter interbreeding. In the 20th century, geneticists started to recognize several principles of Darwinism, referred to as neo-Darwinism. These principles created a better understanding of evolution, a field with ongoing research. The resulting comprehension has supported scientific progress in genetics, such as comprehending the complexities of the human eye and employing the Hardy-Weinberg law to explain its genetics. Neo-Darwinism, also known as the modern synthetic theory, identifies 5 major factors influencing evolution: recombination, mutation, natural selection, gene flow, and gene frequency changes. It is an updated version of Charles Darwin's idea of natural selection, incorporating the research results of Gregor Mendel, population genetics, and molecular biology. This new interpretation of the mechanisms of evolutionary change has significantly improved our understanding of the power and possibilities of evolution. Nothing in biology makes sense except in the light of evolution. This quote from Theodosius Dobzhansky perfectly captures the importance of genetics in the 20th century. This relevance is further emphasized when we look at how listening to music can impact the human brain and positively influence mental well-being. Let's dive into this fascinating topic. Biological isolation, sometimes referred to as reproductive isolation, is a key factor in speciation. Proposed by Dobzhansky in the early 20th century, it indicates that 2 distinct populations of a species can be prevented from interbreeding due to a variety of biological factors. These can be split into 2 main groups. Geographical isolation, when 2 populations are separated by distance, and reproductive isolation, where differences in either behavior or structure prevent mating. This is then further subdivided into premating isolation, when the 2 populations can't mate, and postmating isolation, where hybrid offspring are not viable. Isolating mechanisms take many generations to build up and can be a major contributing factor in speciation. Geographic and reproductive isolation are fundamental in genetics. When populations are separated geographically, they lack gene exchange. Reproductive isolation is established when populations are hindered from exchanging genes through one or more isolating mechanisms such as pre-mating or pre-zygotic ones that avoid zygote formation, or post-mating or zygotic mechanisms that reduce hybrid viability and fertility. Comprehending these 2 concepts is essential to understand genetics. Pre-mating and prezygotic mechanisms, like ecological isolation, temporal isolation, and sexual or ethological isolation, all work to hinder the formation of hybrid zygotes. Ecological isolation occurs when the populations in question are in different habitats but in the same area. Temporal isolation takes place when mating or flowering happens at different times. Lastly, sexual or ethological isolation is when mutual attraction between members of different species is weak or non-existent. Hybridization is still a crucial way of exploring genetic diversity. Even though hybridization can take place in the wild, Dr. R.C. Punnett uncovered isolating principles that arise post-mating, as dubbed zygotic isolating mechanisms, that reduce the fertility or strength of hybrid zygotes. Hybrid unfitness is applicable when referring to the limited strength of hybrid zygotes. Hybrid sterility occurs when F1 hybrids from either sex or both sexes lack functional gametes. Furthermore, hybrid breakdown is the decreased fertility or viability of F2 or backcross hybrids. Population is an important concept in genetics and is key in understanding the Hardy-Weinberg law of equilibrium. Population is used to refer to a group of organisms that have a common trait, typically inhabiting the same geographical area. There are 2 types of populations. Allopatric populations, which are geographically isolated from each other, and sympatric populations, in which 2 distinct populations exist in the same area and evolutionary speciation takes place.